Good afternoon. We're going to do a demonstration on molecular modeling, and we're going to actually look at the structure of invertase, and we're actually going to do some molecular docking today. So this is the structure of this is our website. So that this tutorial is actually the molecular modeling of Beta-Fructofuranosidase. And so that's an enzyme that catalyzes hydrolysis of the glycosidic bond of sucrose into glucose and fructose. It's actually a heavily used enzyme in the confectionery industry and in food chemistry. And it's very important, obviously, because living organisms need glucose as a source of fuel. And so that's very interesting. So we're going to look at that. We're also going to model the structure of sucrose as well. You can follow along our site. This is the direct link to the site right here. And we've also got additional tutorials on the site as well too. So you can actually follow along in that tutorial if you wish, right? So when we get started, we're going to use the software Mo 2024. So this is by Chemical Computing from Montreal, Canada. And this is a very good this is a very good software, and it's very user friendly, and so I recommend using it. It is not free, unfortunately, but it's kind of like it costs the university about the same as Microsoft Office on a per user basis. So it's like Microsoft Office for molecules, if you will. So it's very economical software and very popular with our students. So let's go ahead, and the first thing we need to do is model sucrose. And so I'm using an Ubuntu Linux system right here. When we launch Mo, I'm just going to type MOE at the user prompt, and that launches our software. And this just tells us about some of the new features in Mo. And now there's two ways to import sucrose into the system. Of course, you could go to PubChem. I've already got a video showing how to download a molecule from PubChem and import that. What I kind of like when we teach this tutorial to our students in our biochemistry laboratory courses, I like to teach them how to use the builder software to build a molecule from scratch. And that also has the advantage of building that molecule with, everyone kind of has their own ways of building things, so it has the advantage of teaching students a little bit about different conformations and different ways to energy minimize the same general molecule. So there's a little bit of natural inherent entropy in that process as well. So we can click on the builder right here. Now when the builder, this is the builder tab. I like to attach it to the side, because if you don't do that, then if you're flipping between the center screen and the right side, it can get, it's not the, it'll put the builder behind the molecule and they have to move it out of the way. So I like to attach it right to the side here. And so our structure of sucrose, we need to read the structure of sucrose, which is provided right here in our tutorial. And this is the 2D structure. Remember from organic chemistry what wedges and dashes mean, right? Every time we see a wedge, that means that bond is coming towards you. And everywhere where we see a dash, that means the bond is going away from you. There are two rings in sucrose. There's a glucose ring, that's on the left here. There's a fructose ring on the right. And they're separated by a alpha 1 to 4 glycosidic bond, actually an alpha 1 beta 2 glycosidic bond in the center, right? The alpha is because this oxygen is going down in the structure of glucose. So we want to kind of remember that. And the first thing, now when we're building a molecule, we've got carbons, hydrogens, and oxygens. We're going to sketch the carbons. I'm going to click on this ring over here. I'm going to give this molecule a name and call it sucrose. And just to make it a little bit easier to view, I'm going to give myself a ball and stick model. And if we kind of put the molecule a little bit on the side, that way, you know, if we're straight up and down, sometimes it's more difficult to see which is forward and which is back. So here, if we look at this top atom, this hydrogen's coming towards us, that's a wedge. This hydrogen's going away from us, that's a dash. So on the top, this hydrogen needs to be a carbon based on our structure, and that's coming towards us. And I'm going to click the oxygen here, that's going away from us, right? A couple things about getting used to the mouse control. If you press and hold the middle mouse button, you can kind of spin the molecule like this, right? If you press the shift key, you can move the molecule like this. Now, if your molecule gets way off the screen, you can always click the center button and bring it back. And then the right mouse click will bring up a menu where you can control different things with atoms and colors, a variety of different controls there. All right, so now let's look at this carbon, and based on our picture, the hydrogen going away from us needs to become an oxygen. The next carbon, this one becomes an oxygen. Then this one. And then I'm going to make this an oxygen for now. And then this becomes my ether oxygen in the glucose molecule. So now basically I have glucose, right? Now, this right here needs to become my glycosidic bond. So that's where I'm going to attach fructose. Now, in order to attach this, I can spin this bond like so. I'm just going to spin it like that. And then this is where we're going to attach our fructose ring. And that's a 5-membered ring, so I can click on this 5-membered ring here and attach sucrose. Now I want to orient my molecule in such a way that I'm going to look directly at the fructose, right? And I want it, so here's my glycosidic bond, here's the attachment carbon. This hydrogen needs to become a carbon with an oxygen. This bond right here becomes my ether oxygen. This becomes another carbon with an oxygen on it as well. This becomes an oxygen, and then going away from us. One of these is coming forward, the other's going away. So there's my sucrose molecule, and that should be the correct stereochemistry. Of course, it's not been energy minimized, so step 1 here, perform an energy minimization. We're going to use the amber EHT force field to energy minimize by going to compute and energy minimize, right? So we need to click down in the lower left-hand corner. There's amber EHT. We fix charges. And we always want to see this molecular system appears parameterized. If we don't see that, Mo is going to guess at the correct parameterization. And you know, Mo does things really good. It's great, but it's not an AI engine, and it's not intelligent or artificially intelligent. So we don't want Mo to guess. So we want to make sure that we have all of our charges applied. We come up to compute, energy minimize, and when I energy minimize, now this gradient, this is 0.1 kcal per mole per angstrom squared. This is kind of like looking at, you know, when we minimize, we're at the top of a hill, we're going to the bottom, right? We're minimizing, so we're going into the valley, right? And the gradient tells us the slope of the hill, right? It tells us the slope going down. If the slope is closer to 1, we're pretty steep. It's like skiing downhill. If the slope is close to 0, we're more horizontal. So the closer I get this gradient to 0, like 0.01, or even 0.00001, that's pretty darn close to 0. That means we're at a fairly horizontal point, the trough of the valley. I usually set it now. If we set down to this level, 1 times 10 to the minus 5 power, it's going to take forever to minimize. That's going to take too long. So I usually don't do that. I usually find that 0.01 is sufficient. And then we just hit OK, and it minimizes. We can then look at our SVL. This is kind of the console. This gives you a summary of everything you've done from, for the beginning. So here, this tells you our force field. This tells me I've gone through 34. Well, actually, I've had 538 energy evaluations, but I have changed the structure 34 times to a lower energy, right? And so the gradient, now this is in scientific notation, so your energy right here, this is 1.6 times 10 to the second power. I guess rounded to 2 to 3 significant digits, it's 1.70 times 10 to the second power. This is relative force field energy. It's not free energy. But when we're comparing two different structures, it's sufficient to compare different structures. Here's our gradient. This is less than 0.01, again in scientific notation. And the root mean square deviation tells us how much we've changed since the starting structure. So the starting structure compared to 0, we've gone to 0.973. So we've moved some bonds around. So that's useful. Now the nice thing about this SVL window here, if you're a good scientist and you record a laboratory manual, you can simply cut and paste this. If you're using like a Jupyter notebook, for example, you can cut and paste this in. And so this is, Mo makes it fairly easy to store your notes, right? So that's always good. You can also take a screen capture, and you can just save the picture. We can just call this Sucrose. Whoops. It'll automatically put a .png in for us. So we've saved that. And sometimes I like, on a black background, we don't like to model on a black background because that gives us more eye strain. But when we're doing, when we're printing things out, all that black is going to use printer ink. So we can go to standard white, and then we can save our picture. There's our sucrose model right there, right? And so that gives us an idea what sucrose looks like at this particular conformational state. But I don't like to keep it in a white background for long because, again, that gives us more eye strain. We like to work as safely as we can, and that, most people think that, inherently, computational chemistry, you don't have to worry about lab safety. Well, in fact, there are certain things you do need to worry about in a computational lab in terms of safety and proper ergonomics, right? Make sure you have a nice, well-lit room. Make sure it's comfortable. Make sure you minimize eye strain. Make sure you get up and exercise every once in a while, too. You don't want blood clots or anything. All right, so here's sucrose. Now, one thing you'll notice is blue dotted lines here, dashed line. That's between an oxygen and a hydrogen, so that's actually a hydrogen bond. We have an internal hydrogen bond stabilizing our sucrose molecule here. So that's kind of nice there. Now, it puts the hydrogen bond from the hydrogen to the oxygen. Whenever we measure a hydrogen bond, we always measure from the heavy atom to the heavy atom, right? So if I measure this distance here, whoops, measure distance. There we go. measures. I need to change distance. That's what I was doing. There we go. There's our distance. So I've measured the heavy atom to heavy atom distance. That's 2.6 angstroms. Any hydrogen bond should be less than 3 angstroms from heavy atom to heavy atom. We never measure the hydrogens, and that's an artifact from X-ray crystallography, as crystallographers do not see hydrogens. Now we want to save this, so let's go ahead and save as sucrose. We're going to save it in Mo format, so now that is saved, right? The next thing we want to look at is our protein visualization. And so we're going to go to the protein data bank and download the protein structure for the yeast, which is Saccharomyces cerevisiae. That's baker's yeast or brewer's yeast. And so we're going to download that PDB coordinates. We give you the coordinates here for EQV. So the protein data bank is rcsb.org. And so here's the protein data bank. You'll notice there's over 200,000 structures in the database, also some more predicted by AlphaFold. And so you can simply search by 4EQV, and that tells us this is the structure of invertase from Saccharomyces cerevisiae expressed in E. coli. This is an X-ray diffraction file, and it's 3.4 angstrom resolution. So it's a good resolution. It's not great, but it's sufficient for our purposes, and so it does have everything we need. We can also look at the paper for this, and I'd highly recommend to go ahead and check this paper out. This tells you information about the protein itself. It also tell you information about where like the catalytic residues are and that sort of thing. So this is useful information, right? And then there's other stuff in the PDB. Now the easiest way to import the file is to go to Mo and just go to Open, go to PDB, and just type 4EQV. Mo has an interface directly to the protein data bank. And so there is our invertase structure. And this is a pretty large protein. It looks very large. If we look at the sequence editor, we can see that we have several residues. Now when I look at residues, I usually like to go down here and toggle between single and 3-letter codes. I find it personally easier to look at the 1-letter codes instead of the 3-letter codes. The first thing we'll notice is all 8 of these sequences are identical, right? So that's basically a homo-octamer, right? So we've put all these sequences. All we need is one of them because they are identical. We're going to highlight B through H and delete these. All we need is that first one. The other thing we see is these W's, which before I went from in the 3-letter representation, it was HOH. You could probably guess that's probably water. Now it's W for water. If we just if we show all the atoms, we can see a whole bunch of little oxygen atoms in here scattered throughout. And these are actually crystallographic water molecules, right? We don't really need these, so we're going to do our docking without the presence of water, so I'm just going to make these go away. Now, the reason there's no hydrogen atoms is because it's a crystal structure. There's actually no hydrogens anywhere in here, right? Because X-ray crystallography does not see hydrogens. But we can add those fairly easily, and all we really have to do, really just add the force field, because you're going to add hydrogens here, And then we're going to fix charges, make sure we have our amber EHT force field. I should have done that first and made sure my force field works. So fixed hydrogens for fixed charges, make sure it's parameterized. That adds protons. Now it added protons based on geometry. It didn't add protons based on where we expect the protonation state for the side chains to be. So to fix the side chain based on pH, we need to run protonate 3D. And so this considers physiological conditions. And so I'm going to use a temperature of 310 Kelvin, pH of 7, and 0.15 salt concentration. And then I usually keep all these parameters the same. I just hit OK. And now we're going to consider the pH and protonate our protein correctly based on the physiological conditions of the protein, right? So now that is done. All right. So we've kind of prepped our, well, before we get here, whoops, there we go. And go ahead and go to File, Save. And I'm going to save this as Invertase. I'm going to save it in low format first. I'm also going to go ahead and save this in Protein Data Bank format, right? We'll go ahead and save that as well. The reason I did that is because we want to try and consider the binding site of our protein, right? And so we're going to use a protein tool called the Protein Frustratometer, right? And so this tool is based on energy landscape theory, and it calculates a protein's potential energy surface based on that energy landscape theory, and then it quantifies the degree of local frustration in residues. And these frustrated sites are often correlated with binding sites for small molecules or protein-protein interactions. So we're going to look at that. Now the link to the frustratometer is right here in the notes. Here's my frustratometer. And that, okay, that was weird. So let's go to Google. You can just go to Google and search for Protein Frustratometer, Frustratometer server, and bring up the Protein Frustratometer. Here it is. So I guess that other link. Not sure what happened there. Okay, so the frustratometer runs, to give you a little bit of background on energy landscape theory, so this is a statistical description of the potential energy surface. And so when proteins fold, we adopt a tertiary 3D structure of that protein. And, you know, proteins are, of course, polymers of amino acids, and so when we're at the top of this funnel-shaped energy diagram, the top of this is very high in entropy. We have a lot of degrees of freedom. Remember, we have a phi and a psi bond angle, and we have a lot of side chain positions. And as we fold, the protein, the conformations narrow in energy down this funnel. And you'll notice the funnel is not smooth, it's very rigid. There's a lot of local minima along the way. And even at the bottom, the native state where it says conformational substate, there's still a lot of local minima down here as well. And so what that means is there's some residues that are not as favorable in their docked conformation, right, in their final conformation. And so these residues have not adopted their lowest energy structure, probably because they're waiting to bind to something, right? So to capture this, we can select a local file. I'm going to take, where's my Invertase? So Invertase, it accepts PDB format, so make sure you upload the protein data bank file. I'm going to put my email address in here. Whoops. And a job description, yeast, Invertase, chain A. There we go. And we'll hit submit. Now the next page, it will bring up a JML, a JavaScript file, so we can kind of just confirm, yep, that's Invertase, chain A. I want to click on electrostatics and apply that. I'll take the defaults, and we will hit submit. And then this maybe take about 10 or 15 minutes, and as soon as it finishes, we will get our structure. We have now finished our protein frustratometer calculation. And so when we look at this, we can see local frustration mapped in our structure. And so it gives us a frustration index that measures how favorable a particular contact is relative to sets of possible interactions. And so this is normalized using the variance of energy distribution. For initial inspection, the server classifies initial contacts as to their high frustration index as minimally or highly frustrated, green and red, right? And so we can see a cluster of red right over here. So we have a highly frustrated cluster over here. That may be significant. Now this is, we can't really do anything here. We can click to show the 3D structure if we want. What I'm going to do is I'm going to go down to the bottom here, and this we've got configurational and mutational frustration. The one most relevant that I look at is the configurational frustration, because we're looking at different configurations of our protein and our amino acids. So we want to look at the configurational side. And this is essentially a frustration density percentage that is 0 to 1. If you multiply by 100, that's 100%. The minimally frustrated residues are the green at the bottom. At the top are the ones we're interested in, because these are highly frustrated. These are likely the binding sites we're interested in. If we kind of draw a line right about 0.8, that's 80%. Well, it's actually 20% highly frustrated. So if we draw a line right across this line here, we can see it drops below or it goes above 20% several times. And we're interested in mainly these clusters of residues that are highly frustrated. So let's download our results. This downloads as a tar file, or an RIR file rather. And this gives you all the information that you have. I'm interested in frustration data. And the file that contains our information, we want the configurational frustration. It's this 580 ENS file. This is just several numbers. I think this is today's date in there and probably the timestamp. So we want this file right here. And I'm just going to click extract. And I'm going to extract that to my home directory. And so this shows me all of my files. We're done with the RAR file for now. And here's my file. Now I'm going to just rename this frustrated data.dat. And we'll just call it that. When I open this up, this is a CSV file. It doesn't look very informative, but you'll see all of the numbers is delimited. It's delimited, space delimited. So it's essentially a table. We could also open this up in Microsoft Excel, and we could actually look at that, right? Now, if we open that up in Excel, I don't have Excel on here. If I do search for Excel, I have LibreOffice. And I can certainly do that. If I drag this over to LibreOffice, I'm going to get a window that, so far, it kind of shows me what I want to make sure is click this space delimited so I can line everything up, right? And then everything's lined up so we can see the residue number in the first column, the chain, it's all chain A. And then we have density. What we really want is column G. This is relative highly frustrated density. And so everywhere where these numbers are above 0.2, well, this is 0.19, that's kind of close. This is 0.31. We want to identify all these residues. Then we could simply plug these in, but I've written a script to do this for us. And so let's, I have a Perl script called get frustrated residues, and this is going to parse our file for us. Perl is a very useful file to parsing data, and you can see my information here. To run this script, I've got a comment in here, and we just type /get frustrated residues.pl. We give it 2 arguments, the name of our frustratometer data file, and our output file name, right? And then it reads those into a temporary array. It implements a counter. The shift-argv file, these actually is what reads the information in. And then we open our file names. Most of the good work is done through this while loop right here. And so if header is 6 in the array, this is where our highly frustrated data is. And if that's greater than 0.19, we calculate a frustration percentage. I'll calculate by 100% for us, and we'll print that to the output file, right? And then we just increment our counter, and we write the total number of residues in the protein. So it's fairly straightforward. To run this, I just run this at the command line. .get frustrated residues. And where's my frustrated data? Frustrated residues.txt. So I've got 2 arguments here. You can actually kind of see them right here. And then we can just open up in gedit our frustrated residues. And these are all the highly frustrated residues in the protein, right? Now then what I need to do is map this out in Mo, right? And so the easiest way to do this is to kind of put Mo over on the side here. And to identify them, I really identify them by by coloring the ribbons. Now right now the ribbons are colored by secondary structures, so this is mostly a beta sheet protein, but there is a few little alpha helices in red. I want to change all this. I'm going to take the ribbons and I make this a fairly neutral gray, because we're going to color in various residues by their frustration, right? And so if I select residues, I'm going to select them in the sequence editor down below, right? So the first thing I want to do is find residue 10. That's right here. And so if you select that, that's leucine 10, then proline 23, then 75, that's a lysine, 83, there we go, and so on. And if I kind of right click and go to residues, and if I just color all these in green, they're going to show up in green down below. And if I select ribbon green, it's going to color the ribbons green. See, these are all in green right here, and these are in green, right? The reason I color them down below is if you're kind of going through, and if you click elsewhere, it's going to deselect everything else. So I kind of like to just make sure we record, and then we can print our data and analyze that easier, right? So we're going to go through this whole list, and we're going to color all of our frustrated residues and identify them. All right, welcome back. So now I have finished labeling my frustrated residues. So these are all the frustrated residues I have in the protein. And so I'm going to go ahead and make sure I save. And we're going to save this as an Invertase. Let's do it all in lowercase. Frustration. Oops. There we go. And so that's important to make sure we save along the way. We've got a little bit. We've got some up here. There could be some protein-protein interactions there. There's a few. There's kind of a little tunnel through the protein, which is kind of interesting over here. And there's some frustrated residues along the way over here. So that could be fairly significant, right? So we're going to look at that a little closer. The next thing we're going to do, of course, one method is usually not quite sufficient. So the other method we're going to look at is something called Evolutionary Trace, right? And so this algorithm performs a multi-sequence alignment and correlates this information against the evolutionary tree, right? And so this only needs the PDB code. So let's go up and load Evolutionary Trace on this website here. And apparently my links are doing weird things. So let's go to the server. When in doubt, just go to Google. There we go. That looks a lot better. Okay. And so this tells you a little bit about evolutionary trace, and it ranks amino acid residues in the protein sequence by relative evolutionary importance for when that structure is available in the protein. And so we can get an idea. Obviously, if things are important, something must be happening. So let's put our PDB identifier in here. Let's hit submit. And there's two ways we can look at this. The first way, here's our trace over here. And well, there's usually a report maker, but unfortunately right now we don't have that available, right? And so we can download our data files here as a zip file. And when we look at this, all the data, so it does a blast search, so it compares, this is how it gets its multi-sequence alignment to compare against other things, and then it looks at the entire evolutionary database. Its ranks file is done here under ranks sorted, right? So again, this file down here contains all the ranks, and this is just a text file, so I'm going to extract this, put this in my home directory, and then again, this is just a simple file. Now I don't have a program that's going to interpret this, but let's just look at what this data shows us, right? Here's the alignment number. Here's the residue number in the protein. So this is the same residue number that we looked at with frustration. This first one is an arginine. The second one is a tryptophan. The coverage is the relative importance position of each residue, and low coverage implies evolutionary importance. So we want this to be low. And then the variability, this shows the residue variation. So right now, these first two residues, it's only arginine, and the second one is only tryptophan. For the third residue, it's either aspartate or asparagine, right? And then the next one is only glutamate or glycine, right? And then the score is this last one, so it ranks it by score, right? And so we kind of want to get the top residues. Obviously, you see as you scroll down, the ranks get much higher. They go up to 119.75. So these scores are kind of like golf. You want your score to be low, right? So we can kind of look at the most important residues in the protein. And so here they are. I usually like to kind of find the most important top 100. And so what we can do is if I just kind of nuke this out of the way and then go ahead and save this, I can then open this up in LibreOffice, and there we go. I need to find my home directory again, and where's my ET? Maybe it is under downloads. ET, yeah, ET rank sorted. There they are. And again, so we kind of want to merge delimiters, and so now they're all sorted out. And so now I've got an easy way to sort these out. And what I like to do is find the top 100 residues in the protein. So if I go down here to row 100, let's go to 101. Then I can delete everything else because these are not important, right? So everything else, I think there's 500 amino acids in here. The top 100 are the top 25%, or the top 20% of the residues in the protein, right? And so now what we can do is we can do a quick sort now. If I try and plot these in Mo, you know, this starts with residue 150, 186. I like to sort by column C because then I can kind of just go straight through from residue 1 to the top. And so let's just highlight all of our residues, data, sort. And I'm going to sort by column C, and I'm going to do it in, I think, descending order. No, I want to do ascending order. I always do that backwards. There we go. So now they're sorted in ascending order. And so they're not sorted by the score. You know, this isn't the most important residue, but they're sorted in order of the residue number, right? And so then we can identify these in Mo. So let's go ahead and do that. If I bring my sequence editor, I can say, okay, residue 8 and residue 9, and then residue 11, right? Residue 20, 27, and 28, 29, 30, 32, 33, 46, 48, 49, 51. We have 52 and 53 right next to that. So then 86, 95. Now 95 is also frustrated, so I'm going to hold off on that. I'm going to color all these in. Let's make these all red. And we'll go residues red, and then ribbons, I want to color those red as well. So that colors everything in red. It colors everything down here. And then that glycine, this is both. So let's make this like a nice bright blue. That's if they're both highly frustrated, right? So that's in blue, and I think I've got a nice blue in here as well, right? So we go through this list and we'll be able to highlight all of our residues, right? All right, welcome back. Now you can see I've got my yeast invertase colored by frustration and evolutionary trace. We can kind of see our sequence editor. Now if I want to look at this data, because we like to report our data in our notebook, I can go to print. Now when I print this, I can print the sequence on a standard 8.5 by 11 sheet of paper, but it doesn't have all the residues highlighted. But what I can do is I can color those residues, and now they're highlighted, and then I can print this out to a standard printer and put that in a notebook, right? And again, as before, we can go to standard white and color this in, but again, we don't want to stay in white. So what we see is basically on the left side of this protein, not very interesting. There's not a whole lot going on. Everything's going on the right over here. So I would think that somewhere right in this area, this is the active site, because there's a lot of frustration. There's a lot of evolutionary important residues over here, right? Now, remember when we looked at that paper, right, in the protein data bank. Yep, here we go. Remember this paper? And if we kind of click on it, you should read this paper, because this also tells us a lot of information. It also tells us a lot about specific residues that are highly important. And if you read this in detail, this also tells you that residue glutamate 203 is very important. And in fact, glutamate 203 is one of the most important residues in invertase. That's actually the catalytic residue. Right here is glutamate 203. And there's only two variations, right? It's either a glutamate or a glycine, and its importance score is 1.7. So it's got to be important. Definitely the catalytic residue. So if we kind of find that, let's locate glutamate 203. That's right here. No, there's 204. There's 203. There it is. So I'm going to kind of highlight that. Go to atoms. Let's make sure we can know what we're looking at. That's right in the middle of all these important residues, right? So I would say glutamate 203 is pretty important, right? So we're going to target our docking centered around this glutamate residue in our protein, and that's where we're going to try and direct our sucrose to. So first of all, let's go ahead and just save this. Invertase frustration ET. I like to save it just to make sure we've got everything worked out. And then we're going to put our force field on. It's parameterized. I want to make sure my amber EHT is working. And then we're going to open up the sucrose molecule that I had previously saved. Right? Now, your sucrose, when we open that up, sucrose will be here, but it's not going to be anywhere. It's nowhere near the active site. Not sure where it's putting it right now. There it is. Yeah. So here's our sucrose out here. We still have that bond. I can simply select the bond and remove distances. So there's our sucrose, right? Let's make sure that's still parameterized, amber EHT. So we want to make this go into this, and we want to target the residue, this glutamate residue. So let's select that glutamate, right? There we go. I want to, if you click on the ribbon, that selects all the atoms of that residue, right? And so then to do the docking, we are going to go to Compute, Dock, and this shows us what we're docking. Now usually when we have these dialog boxes, I like to start at the top and go down, right? So the first one is the receptor. It says use most systems. I'm going to use these selected residues because I want to select and target the glutamate 203 residue. And then the site is also selected residue. Well, actually, the first one, I can just say receptor and solvent atoms, even though there's no solvent. The second one, the site, I want to make sure and select selected atoms, actually. So it'll select all these atoms that I have selected, and they're highlighted in magenta there. pH4 is for a pharmacophore. We're not going to use that today, and then the density is related to that. The ligand, just make sure that sucrose appears in the big window. When Mo does its docking, it uses two methods. The first is a placement method, and it uses the triangle matcher. So essentially it defines the van der Waals surface of the active site, and it puts little triangles to define that surface. The smaller the triangles, the smoother the active site. And it scores this by London dispersion forces, right? We get 30 initial poses, and then for refinement, we're actually going to use the induced fit method, because instead of rigid receptor, we want to make sure the receptor moves around a little bit. So we use induced fit. This uses a generalized Born scoring method. This is a free energy of salvation based method, so we're going to get a free energy score, and that will be in kcal per mole, and we'll get 5 docked poses. And then for our output, I like to just say Invertase Sucrose, and this is going to save the docked poses in an MDB, molecular database file, where we can then retrieve that for later use as we want, right? Once we have all of our parameters in, this is where the fun part comes in. The students love this. I like to kind of put this over on the side because once I hit run, we're going to see a little molecular ballet, and it's going to start dancing around. There it goes. So there's our sucrose adopting as many possible conformations in the substrate as it can. Eventually it'll stop dancing around, but we'll get in the molecular database on the right side of the screen, it will populate with the top 5 poses right there. You can also see several hydrogen bonds that are being formed in the residue as well as it forms. Remember, when we dock a ligand to a receptor, most of the time it's not going to form a covalent bond. It likes to form a non-covalent interaction, either hydrogen bonding or van der Waals hydrophobic interactions, because then that makes the enzyme reversible, so the enzyme can then be reused for other things. So we just got our top 5 docked poses right here. Now this is one of those things where when we teach this in our biochem lab, our students love to just print this whole table out there, and they think this is all of our data. You know, students love data, and that's an important thing, but you want to record the important data you see. Some of these columns, like R-seq, that's the index of the receptor, M-seq, index of the ligand. This is just one. That's not really all that interesting. Again, mole. Well, we've docked sucrose 5 times. Receptor. There's our 4-EQV. That's our PDB score. All we really need is to record we've got 1 through 5 docked poses. And then the S, if you hover over this, it kind of tells you what it is. This is our final docking score. And it tells you the scoring method is GBVI, WSA. That's our generalized Born docking method. And so that gives us a solution, a docked free energy of binding, and that's delta G in kcal per mole. So that's minus 7.5 as the top, going down to minus 6.5 as the bottom. We do have like an RMSD. This tells us the RMSD of the ligand before docking and the final pose. We've considerably moved around. We've refined an RMSD. We have some other scores. Now, S is the same as E-score 2. The E-score 1, remember we did the first docking method with our London DG, and that was our triangle matcher, so that's E-score 1. And then E-score 2 is the docking method with the generalized Born, which is recorded as our docking score S right here. And then these are just, I don't really worry about those last ones. To load this, all I do is I go to right-click on sucrose number 1, send to Mo. Definitely don't want to clear the system because then it'll put the sucrose in there and clear the protein. We don't want that. So here's our sucrose. Now, if it's kind of hard to see with a lot of other atoms in there, so let's just double-click on the atoms. We can make this a little more visible here. And there's our sucrose, and it happens to be located in close proximity to our catalytic residue right at the middle, right there, right? We also see several hydrogen bonds down here with this hydroxyl of the fructose part right there. And that's sitting between like probably maybe that's a glutamine and a histidine over here. So we've got others. Now, it's kind of hard to see with all these other atoms, so let's double click on here. Now let's go to compute and ligand interaction. This is going to give us a ligand interaction map. So this is a 2D representation of our sucrose in our docked conformation. Now, when you first bring this up, this is ligand number 1. This is the sucrose that's way outside the active site. So this is not what we want to look at. We want to look at ligand number 2 and apply that. This is in the active site because now we see various residues. We see them colored by the green residues are greasy, that means hydrophobic. The pink residues are polar and some are acidic if they're highlighted in red. If they're highlighted in blue, they're basic. We see a couple specific interactions like hydrogen bonds. Down here we see these are all those residues right next to our hydrogen bonds. That was actually a tryptophan that I was misidentifying as a histidine. I did get the asparagine right? We've got a glutamine nearby. So this shows us in the active site. We can also print this out if we wish, right? We can export this as a PNG file. So we can import that into a laboratory notebook. And we can also isolate, right? If I click isolate, well, then we see the 2D representation as well as the 3D representation right here. And then we can kind of look at everything in 3 dimensions, and it's also labeled, and only the residues in the active site are actually labeled. So it doesn't put everything. And, then if we want to print this out, finding the right orientation, I'm going to try and See, if we print this out, we've got a lot of wasted space where nothing's happening. So we want to kind of zoom in and minimize that wasted space and make sure that we can visualize our actual protein. And then don't forget when we print to go ahead and go to a white background for printing, right? So that is our doc. Now that's just doc confirmation 1. We do have a few more doc confirmations, but in the interest of time, I'm going to leave that to you, right? And we've kind of finished now. I've developed this as a tutorial that you can go to our 3D biochemistry website and look at. I've also put several questions along the way for you to, if you're a teacher, you're an instructor, you want to ask, you want to put this as a student, feel free to go ahead and use this. And if you find this helpful, go ahead and comment, leave a comment below on the file. You can click the like button, and if you like a lot of these videos and you find them helpful, go ahead and subscribe to the channel, and you'll see more stuff. So again, thanks for watching, and I hope you found this useful.